Good morning. Hello, this is Shahid Naseem once again. And once again, today we are going to talk about some new things. And this is a live session. And I think this is the second time I'm doing this live session. Now, the reason I'm doing these live sessions are to spread more awareness and to talk about these things, to normalize these these things. And today I have some few very important things to discuss with you. So before any further ado, let's get started. So there are these two new researches that I wanted to share with you. The first one is very important, and it is an autism study which unveils new genetic players and role of language delay. Now, language delay is a very important thing. In fact, one of my nephew has this problem. He has this language delay problem. It's not actually related to autism, but somewhat on near to that spectrum. So we're going to discuss about this. So let me share a summary of this first with you. Researchers shed new light on how genetics may influence the development of autism spectrum disorder. Now, the study, the largest ever to focus on families with multiple children diagnosed with autism, has revealed 7 potential risk genes. Additionally, it has provided uh, genetic evidence for reconsidering language delay as a key component of ASD. These findings highlight the complex interplay between common and rare genetic variations in determining autism risk. And also, some key facts about this research is that, let me share those key points. UCLA, this is the research is from the UCLA. UCLA health researchers conducted this largest study to date on families with more than one child diagnosed with autism, uncovering 7 potential risk genes. This is very important. The study also highlights the importance of the role of both common and rare inherited genetic variations in autism risk. It also reveals that genetic evidence from the study suggests that language delay and dysfunction may be a core component of autism, contrary to the current DSM-5 diagnostic guide. And I've already told you that the source of this research is UCLA. UCLA was the place where this research has been conducted, and this research has been conducted by UCLA. UCLA health researchers have published the largest ever study of families with at least 2 children with autism, uncovering new risk genes and providing new insights into how genetic influence whether someone develops autism or spectrum disorder. Sorry. Now, this new study published in the Proceedings of the National Academy of Science also provides genetic evidence that language delay and dysfunction should be reconsidered as a core component of autism. I've already told you this one before. Now, most genetic studies of autism have focused on families with one child affected by the neurodevelopmental disorder. Some families with multiple affected children, the role of rare inherited variation or its interaction with the combined effect of multiple common genetic variations that contribute to the risk of developing autism. Now, study design is very critical, and not enough attention has been paid to studying families with more than one affected child. This is said by study author Dr. Daniel Gashman. I'm not sure if I'm pronouncing this word correctly or his name correctly, but the first one is good. Dr. Daniel, that's his name. Now, the Gordon and Virginia McDonald Distinguished Professor of Human Genetics, Neurology and Psychiatry at UCLA. So this is the one, the guy, the doctor who done this research. Autism is very highly heritable. It can be inherited from your parents or from your grandparents or throughout your family life. It is estimated that at least 50% of genetic risk is predicted by common genetic variation, and another 15 to 20% is due to spontaneous mutations or predictable inheritance patterns. The remaining genetic risk is yet to be determined. Now, having said that, for this study, researchers performed whole genome sequencing in 4,551 individuals from 1,004 families with at least 2 children diagnosed with autism. This group included 1,836 children with autism and 418 children without an autism diagnosis. The researchers found seven potential genes that are predicted to increase the risk of autism. They are the genes are PLEKHA8. I'm sure you won't be interested in names. I'll link this study. I'll share the link of this study in the description, so you can go there and actually read the whole study if you're interested in that study. You find the genes, name of the genes there. This is remarkable. This whole study is very remarkable because other studies have had to analyze much larger cohorts to identify a similar number of novel risk genes. This is because in this case, most of the new genes were supported by rare inherited DNA variations that were transmitted from parents to children with autism. The researchers also examined polygenic risk, in which a combination of commonly found genetic variations can raise the likelihood of developing autism. They found children who inherit rare mutations from unaffected parents in combination with polygenic risk are more likely to have autism. This help explains why parents who carry a single rare mutation may not show signs of autism even if their children do. It also lends support to the liability threshold model, a concept in behavioral genetics that holds there is an additive effect of genes that influences the probability that someone develops a certain trait. Now, in another important finding, children also had children who had language delay had a higher likelihood of inheriting a polygenic score associated with autism, while there was not a similar relationship for children without language delays. This pattern was specific to autism and was not seen in other traits like educational attainment, schizophrenia, or bipolar disorder, suggesting there is a link between the genetic risk for autism and language delay. However, the most recent edition of the professional guidebook used by mental health providers to diagnose disorder, the Diagnostic and Statistical Manual of Mental Disorder, 5th edition, which is usually called DSM-5, does not consider language delay a core autism symptom, citing the variability in language ability among people with autism. The association of a general risk for ASD that was strongest in those with language delay suggests that language is actually a core component of ASD. This finding needs to be replicated in larger cohorts, especially those recruited more recently under DSM-5. This was said by Dr. Daniel Gashman. Again, I'm not sure if the pronunciation is right for this Gashman word, but Dr. Daniel Gashman said this. Now, Dr. Daniel Gashman is a professor, I've already told you, and the study was conducted by UCLA. This was very important, this study that I wanted to share with you, that autism studies has unveiled new genetic players and role of language delay. Now, there was this another study which I wanted to share with you, and this study is also very important, and I've been studying these researches, and I was quite impressive, to be honest. And this one was a study, and this research shows how brain inflammation in children may cause a neurological disorders such as autism or schizophrenia. So if anyone in your family, or even if you or anyone who's in your surrounding has autism or any kind of disorder, this is very important for you to listen. So pay attention, please. Now, findings shows that inflammation stops some neurons from mutating in the developing brain, which could open the door to new treatments. Now, severe inflammation in early childhood is a clinical non-risk factor for developing autism and schizophrenia. Now, for the first time, scientists from the University of Maryland School of Medicine has discovered that inflammation alters the development of vulnerable brain cells, and this could have mechanistic links to neurodevelopmental disorders. This finding could lead to treatments for many different childhood onset neurodevelopmental disorders. Using single cell genomics to study the brains of children who died from inflammatory conditions such as bacterial or a viral infection, or asthma, along with those who died from a sudden accident. Researchers from the University of Maryland School of Medicine led a study that found inflammation in early childhood prevents specific neurons in the cerebellum from maturing completely. The cerebellum is a brain region responsible for motor control and higher cognitive functions used in language, social skills, and emotional regulation. Faculty from the University of UMSOM's Institute for Genome Sciences, Department of Pharmacology, and the University of Maryland Medicine Institute of Neuroscience and Discovery conducted the research, and the study appears in the October issue of Science Translational Medicine. It is part of a collection of nearly 30 papers describing the development and diversity of cell types in the human brain. All of these studies were coordinated by brain researcher through Advancing Innovative Neurotechnologies Initiative, Cell Sensors Network, a multi-site consortium funded by the National Institute of Health, NIH. Now, previous research has shown that babies born with abnormalities of the cerebellum frequently go on to experience neurodevelopmental disorders, and animal models exposed to inflammation before birth also develop these conditions. Once again, if anyway you are connected to this is very important for you. As said by Dr. Seth Ament, who is a PhD, we looked at the cerebellum because it is one of the most, it is the first brain regions to begin developing and one of the last to reach its maturity, but it remains understudied. So this was said by Seth Ament, who is a PhD, IGS scientist and associate professor in the Department of Psychiatry at UMSOM, who co-led this research with Margaret McCarthy, who is also a PhD, the James and Caroline Franklin Dean's Professor Chair in Pharmacology and Director of MIT. She said, with the fairly new technology of single nucleus RNA sequencing, we could look at the cell level to see changes in the brains. Now, Dr. McCarthy, she also said that this has never been done before in this age group and in the context of inflammation. The gene expression in the cerebella of children with inflammation were remarkably consistent. The researchers examined donated post-mortem brain tissues of 17 children who died when they were 1 to 5 years old, 8 from conditions that involved inflammation and 9 from accidents. None of the donors had been diagnosed with a neurological disorder prior to death. This is important. Two groups were similarly in age, gender, race, ethnicity, and time since death. These unique brain tissue specimens had been collected over many years by UMSOM researchers at the University of Maryland Brain and Tissue Bank, a tissue repository of the NIH Neurobiobank, as well as the Maryland Brain Collection of the Maryland Psychiatric Research Center. This study found that two specific yet rare types of cerebellar neurons were most vulnerable to brain inflammation, the Golgi and the Purkinje neurons. At the single cell level, these two types of neurons showed premature disruption of their which we call maturation. Okay. As I've said before, the Dr. Armand has a very deep look on this topic and he has conducted this research. He said that although rare, Purkinje and Golgi neurons have critical functions. He also said that, he said, and I quote, during development, Purkinje neurons from synapses connecting the cerebellum to other brain regions involved in cognition or emotional control, while Golgi neurons coordinate communication between cells within the cerebellum. Disruption of either of these developmental processes could explain how inflammation contributes to conditions like autism spectrum disorders and schizophrenia. As with many diseases, both in genetics and environment, in this case, inflammation likely contribute to the risk of developing these disorders. That's why it's crucial, it is very crucial to understand the roles of specific cells within the brain regions, as well as how they interact with the genes to influence brain function. To find treatments for brain disorders like ASD and schizophrenia, as well as others including dementia, Parkinson's disease, or substance use disorder. Now the study still goes on, goes on and goes on. It's a very detailed study, and I would like to share some, like, I want to read the whole study with you, or at least share some of the important aspect of the study. But I think the study is very detailed and very long. It will take a lot of time. So if you're interested, do let me know. Leave me a message or a comment, and I'll share some more interesting details with you, some more interesting researches about autism and ADHD with you. For now, this is it from me. Take care, and take very good care of your people who are with your surroundings. Okay, somebody, Shivaji Khikar and Sayan has commented. Sayan, hi there, I hope you're watching. I'm really sorry I missed your message. And Shivaji, yeah, it is not a disease. Autism is not a disease. Let me repeat myself. Autism, ADHD, it's not a disease to be cured of. It is a condition. It is a condition, and they can work very effectively in society. So it's not something which you can cure or which you can get rid of. Autism is more like a condition, and you have to work with it. We are simply a bit different than us. So it's a completely different debate, and we can talk about this, how autism is, we can deal with people who are affected, who has this condition, this particular condition. So the question you ask is not a question really. So like you have asked that if they can be cured, it's not a disease to be cured of. It's not a disease, it's a condition. So please think about this, ponder on this thought. Autism, ADHD, these are not diseases. These are people who have these condition, who are going through this condition, who have this condition. They are not sick. It's simply a condition. Like different people have different kind of conditions. If somebody is structuring, or if somebody having a speech delay kind of a problem, you know that it's not a disease particularly. It's a condition. So similarly, autism is not a disease. It's a condition, and you can work with those people, how they can effectively live, how can they can be more communicative. You can work the communication. You can better work. I'm really sorry about that. This tongue twisters. I get this sometime. Okay, maybe I also have some kind of condition, but I've never got myself checked. Okay, the thing is, these kind of conditions, you can work with these people, and you can make their life a bit better by working on them every day. Like the mostly in these cases which we have seen personally, I have seen personally, the problem is communication. So you can work to make the communication better, to communicate better, to learn the signs of, if the child is very young, you can learn the signs. What are the signs that can tell you that the child may have autism or ADHD or some other kind of a condition or disorder? So this is not a right question to ask. That's it from today. From other day, I'm gonna talk to you about some different studies, some different researches. So if you think that this is something which is relatable to you, or which you want to talk about, or if there is any other question that you want to ask, please do let me know. You can comment on this video, and I'll get back to you with those questions, and I'll talk about those questions. I'll talk about those topics. I've also this another study that I was reading a few days back, and this study was particularly from Yale. The study was conducted by Yale. Now, Yale scientists revealed two paths to autism in the developing brain. This is very important. This is a very short. It will take a little bit of your time, but this is important. Two distinct abnormalities that arise just weeks after the start of brain development have been associated with the emergence of autism spectrum disorder. Now, two distinctive neurodevelopmental abnormalities that arise just weeks after the start of brain development have been associated with the emergence of autism spectrum disorder. Now, according to a new Yale-led study in which researchers developed brain organoids from the stem cells of boys diagnosed with the disorder. And researchers say that specific abnormalities seem to be dictated by the size of child's brain, a finding that could help doctors and researchers to diagnose and treat autism in the future. The findings were published on, I guess, August 10, in the journal Nature Neuroscience, and said by the Dr. Flora Vaccarino. I think Dr. Flora Vaccarino. I'm really sorry if I'm not pronouncing the word right. Dr. Flora Vaccarino. She said that she said, and I quote, "It's amazing that children with the same symptoms end up with two distinct forms of altered neural networks." The Harris professor in the Child Studies Center at Yale School of Medicine and co-senior author of the paper, Dr. Flora, she said this, and using stem cells collected from 13 boys diagnosed with autism, including 8 boys with the macrocephaly, a condition in which the head is enlarged. A Yale team created brain organoids, small three-dimensional replicas of developing brain, in a lab dish that mimic neural growth in the fetus. They then compared brain development of these affected children with their fathers. Patients were recruited from children colleagues at the Yale Study Center, which conducts research, surveys, and training to improve understanding of health issues facing children and their families. The study was co-led by Alexander Jordan, Finn Wu, and Jessica Mariani, all from Vaccarino's lab at the Yale School of Medicine. And about 20% of autism cases involve individuals with the macrocephaly, a condition in which I've already told you child's head size is in the 90th percentile or greater at birth. Among autism cases, these tend to be more severe. Intriguingly, the researchers found that children with autism and macrocephaly exhibited excessive growth of excitatory neurons compared with their fathers, while organoids of other children with autism showed a deficit of the same type of neurons. The ability to track the growth of specific types of neurons could help doctors diagnose autism, the symptoms of which generally appear 18 to 24 months after birth. The authors said this. The finding may also help identify autism cases that might benefit from existing drugs designed to ameliorate symptoms of disorders marked by excessive excitatory neurons activity, such as epilepsy. Vaccarino said autism patients with macrocephaly might benefit from such drugs, while those without enlarged brains may not. It was said by her. And creating biobanks of patient-derived stem cells could be essential to tailor therapeutics to specific individuals or personalized medicine. So this was said by Amazon's Alexej, an associate professor of biomedical informatics at the Mayo Clinic, is a co-senior and also author of the paper. So these were the most important things which I wanted to discuss with you. There are a few other studies that I want to discuss, but time is very short and I'm already speaking for last, I guess, 20 minutes. So this is it from today. Some other day I'm going to talk about some other few studies. So in the meantime, if you have something to talk about, if you want to shed light on some cases, if you have something as a case which you want to discuss with us, do let us know, leave us a message or email us, and we'll get in touch with you and we'll discuss about these kind of things. Thank you so much for watching. Take care. Bye-bye.